Welcome to Biomentors. In this video, we will discuss some concepts about principles of biotechnology, which is from chapter 11. Okay, so here's the first term, genetic engineering. What is this genetic engineering is? So in simple terms, copying DNA of interest from one organism and paste it into different host organism. How does this happen? Like, what are the steps? So basically there are 3 steps to produce genetically modified organism or to perform genetic engineering. First step is you need to identify which DNA segment you have to take further for genetic engineering. So first is the identification of DNA with desirable genes. Next, we have to introduce these genes into a suitable vector or a carrier to take it further. So next is introduction of the identified DNA into host. Okay, and when it does, ultimately we have to maintain it and introduce DNA in host and transfer of the DNA to its progeny to quantitate its number, means to increase its number in high amount. So this is a diagrammatic figure how recombinant DNA technology can be done. For example, you can see there is a human DNA where a gene is insulin. We have to obtain a certain product from this insulin gene. As you know, the insulin gene is responsible for maintaining blood sugar level, and diabetic patients take it artificially or from outside source to maintain their sugar level. Okay, so we are interested in this insulin gene for now. Now we have to make usable insulin to provide or to give it to the patient as a medicine. How do we do that? Because a human body will just produce the enough amount of insulin for its own use, so we can't use this insulin in a larger scale to use it as a drug. So what we do is we are using this technology for our purpose. So we are using a plasmid vector which is autonomously replicating unit, and we will produce a cut in this plasmid to introduce our gene of interest. Okay, and how we will produce a cut? There is an enzyme which is called molecular scissor in the molecular biology. Okay, this is called restriction enzyme. So we have 3 components here. We have our gene, we have the carrier or a vehicle, let's suppose as an auto rickshaw, which carry you somewhere. So this is a plasmid, and the third is restriction enzyme. Now we have to insert this DNA into this plasmid to get it to somewhere. How we will do that? How we will ligate this gene into this another DNA? There is another enzyme called ligase, which is called as also a glue in the molecular biology. Once we ligate this piece, we need to introduce in a certain alive host organism that can increase its number as copy number, like exact the same gene. So this is also called as gene cloning. Now let's suppose there is a bacteria. Bacteria are very microscopic organism, as you know. They reproduce in a very high amount, like in a logarithmic phase, very high in very high number in very short duration of time. So this is a suitable host for performing this process. Now, as this is called as a competent bacteria, and we have our DNA in the host or plasmid, what we will need to do is to introduce this plasmid in this competent bacteria. Okay, once this plasmid is inserted into this bacteria, what next? Next is we will allow this bacteria to reproduce. What happens when bacteria reproduce? It grows continuously in very large number. Suppose in 24 hours it will produce millions in numbers. As you know, in logarithmic reproduction, it will produce like from 2 to 4 to 8 to 16 kind of. When it will produce in high number, it will serve two process. One, we will get very high number of DNA to perform other experiments, but remember what is the purpose here. We have to produce the insulin, which is a medicine for a diabetic patient. So what we will do, we will purify this insulin. Why purify? So that we will use it in a good way, in a usable form, to provide it to the diabetic patient. So in next chapter we will see how downstream processing is done in the industries. So for now we will just see that we will purify this insulin to make further use. Okay. Now there's some points about plasmid. So as I have told, that plasmid is autonomously replicating circular extra chromosomal DNA. As you see, this is double stranded circular. Okay. So when this present in bacteria, it serves as additional features to bacteria. Like suppose when scientists get to know about this thing, this plasmid, it used to serve as an immune system to bacteria. How? This plasmid helps the bacteria to fight against viral infection. When scientists get to know about its use, they have exploited this segment of DNA to carry their gene of interest or to serve their purpose. Okay, so there are some important features of plasmid for cloning. What? ORI means origin of replication. This site is very important for any gene to start replication, and only when any gene will replicate, when it will have to produce in more numbers, when it reproduce. So this is very important DNA segment. Next is selectable markers. We need selectable markers. For example, ampicillin resistant gene and tetracycline resistant gene, to distinguish between transformants or recombinants between non-recombinants. Third is cloning sites. Cloning site is present within the selectable markers. You see, there are abundant of cloning sites. Cloning sites are the sequences where we insert our DNA. Okay? Now, vectors for cloning genes in plants and animals are YAC and BAC. YAC is yeast artificial chromosome and BAC is bacterial artificial chromosome. There are two examples of plasmids, PBR322 and TI plasmid. TI plasmid is tumor-inducing plasmid, which is present in Agrobacterium tumefaciens, which used to carry genetic engineering in plants. Okay. So next is there's some point about restriction enzyme. What is this restriction enzyme? Why it is called molecular seizure? Because restriction enzymes cut DNA at palindromic sites. Okay, more about this on next slide. So when restriction enzymes scans, how? Like this, it goes, and when it scans a particular sequence of DNA, then it cuts it here. Okay, how? Like I have told you that in DNA or DNA strand, there is nitrogenous base, ribose sugar, and a phosphate group. So exactly here the restriction enzyme produces a cut by hydrolyzing this bond. Like plasmid, it also found in bacteria naturally. Why? It acts as an immune system in bacteria. Thousands of restriction enzymes have been discovered so far and used in labs to perform recombinant DNA technology. One example of restriction enzyme is EcoR1. And EcoR1 is a word in which the terms implies like E means Escherichia, which is genus of E. coli. Then this CO means coli, which is a species name. This R stands for strain, and this number in which it has been discovered. This is a figure I have been borrowed from a research paper just to show you how restriction enzyme produces a cut. So as I have told you, in DNA there is a nitrogenous base, a ribose sugar, and a phosphate group. So it usually cuts in this section between phosphate group and 3' OH group. So this is the usual cut it makes. It produces a free 3' OH group and free 5' phosphate group. Sometimes it also produces a cut like this, but this is rare. Now, why we need genetic engineering or why RDT is important? Because as I have told you, in diabetes sugar level is get disbalanced, so we need a continued source of insulin. And how can we do that? As I have told you also, that a single human or any human or individual can only produce enough amount of insulin for its own body. So we can't take insulin from someone's body. So we need to produce more and more amount of insulin. And how can we do that? By genetic engineering. It's an easy way. I have already shown you. But initially, we used to take this insulin from cow, which is offensive for some people in several ways. Like some people are allergic to it, and some people are offended because of their religion. Okay, so there's an alternate way which has made insulin to produce on a commercial scale, and by reducing the cost by introducing it into bacteria. And so far not much people are familiar what bacteria is, so if nobody knows what it is, they think it's okay to take it from there. So bacteria is also a good source and a cheap source for producing this very important medicine. That's all from my side today, and I will discuss more points from this chapter. And thank you for watching.